So we are focused on a group of high-risk acute leukemias that are collectively known as lineage ambiguous acute leukemias. These are relatively rare, but they have very poor prognosis. And one of the reasons for this is because there are multiple challenges in terms of accurately diagnosing these leukemias in patients because they typically express antigens associated with both the myeloid and the lymphoid lineage. And so that poses challenges for diagnosing and classifying them, and also in choosing the most appropriate therapy. And there's relatively little known about their underlying biology because they're so rare. So this study focused on this collective group of acute leukemias, taking an approach that's orthogonal to looking at cell surface markers to classify them. So we looked at the transcriptional profiles of as many lineage ambiguous leukemias as we could compile, spanning from mixed phenotype acute leukemia, acute undifferentiated leukemia, early T-cell precursor acute leukemia, which is considered a subtype of T-ALL, but these leukemias frequently express myeloid and stem cell antigens. And so by looking at the gene expression profiles of these leukemias, we aimed to better classify them and understand their underlying biology. Sure. So as I said, we took a transcriptional approach. So we analyzed RNA-seq data from, in this pan-acute leukemia analysis, there were over 2,500 samples. And this allowed us to perform various methods to look at, the relationships between different leukemias. So we used T-SNE, projection analysis, and hierarchical clustering to examine the relationships among this entire cohort. And as we expected, the group of B-cell acute lymphoblastic leukemias formed very distinct gene expression clusters that corresponded to genomic alterations. But this group of, the non-B leukemias comprised of T-ALL, AML, and these lineage ambiguous leukemias, we did observe substructure within this group. And, interestingly for us, specifically within the lineage ambiguous leukemias, we identified a new subtype, and I say new just because it hasn't really been described in the literature before, and this is likely due to limits on sample size. So in this analysis, we identified 63 samples that fell within a very distinct gene expression cluster. So the first main result is this identification of this subgroup. All of these samples are defined by having structural alterations, genomic structural alterations that target the BCL11B locus on chromosome 14. We identified this from whole genome sequencing analysis, and in a subset, RNA-seq was able to identify this as well. And the characteristics of these structural alterations is that this leads to expression of BCL11B from only one allele, so the allele that has the structural alteration. But importantly, we found that this group was comprised of multiple diagnostic entities, and this sort of ties back to the challenges that I mentioned in the beginning. So, in the clinic, these leukemias will be diagnosed as different diseases, whether it's T-myeloid MPAL or ETP-ALL. But we found that actually about 30 to 40% of this biologically similar group of leukemias were T-myeloid MPAL, and 35% were ETP-ALL. So this suggests that our ability to identify biological distinctions among leukemias just based on immunophenotype or morphology is insufficient. So this is digging into the molecular underpinnings of this group. So as I said, we identified structural alterations targeting the BCL11B locus in this gene expression group. And these alterations result in BCL11B, the gene. So the breakpoints spare the entire coding gene. I think that's important to indicate. And so this results in BCL11B being placed in other regions in the genome. And because we saw monoallelic expression, we immediately hypothesized that these alterations are targeting BCL11B expression. So they're not perturbing the gene itself, like we do see mutations in this gene in canonical T-ALL. But here we hypothesized that it was what's known as enhancer hijacking, which is essentially where a gene that's normally repressed is aberrantly or ectopically activated because it's now placed in proximity to a strong transcriptional activator. These are enhancers, and there's hundreds of thousands of them in the genome, and they are active in very specific times during development. And so we saw that all of the rearrangements that we observed in this group place BCL11B near a very strong enhancer that's active in hematopoietic stem and progenitor cells. And one of the implications of that observation is, again, supporting that the cell of origin for these leukemias is a stem or progenitor cell, because BCL11B is normally only expressed in the T-lineage. And so this suggests that this alteration occurred before the onset of T-cell differentiation. And so the chromatin topology analysis was aimed to first, prove that these rearrangements place this BCL11B gene near active enhancers. So one benefit of the approach we took, which was called HiChip, is that it simultaneously identifies enhancers by virtue of being able to tell you where H3K27 acetylation is located in the genome. This is a post-translational histone modification that's associated with transcriptionally active chromatin. And it also gives you information on the other regions of the genome that regions with this marker interacting with. So if you want to identify which genes a given enhancer is interacting with, you could use this approach to get at where that enhancer is and what are the genes that it's interacting with. And so, the take-home message is that this approach demonstrated that these rearrangements place BCL11B near active enhancers that correspond to active hematopoietic stem and progenitor cell enhancers. And importantly, these enhancers are forming chromatin interactions, so physically looping to the BCL11B gene. And that provides, kind of the strongest line of evidence that we can provide in primary patient samples that the mechanism of BCL11B expression is through enhancer hijacking. Yeah, that's a great question. So there are a couple of things that we would like to say about that. The first is that we believe that that our results support that taking transcriptional and genomic approaches to better classify leukemias is a very valid approach, and that we need to, continue doing this to continue identifying these subtypes that are present within homogeneous diagnostic entities. So that can lead to better, our better ability to classify leukemias and then start to correlate, these different occurrences with treatment response and outcome. And so that's kind of a first step that needs to be taken. We acknowledge that identification of this alteration is based on next generation sequencing. And so its immediate utility to be to identify this group in the clinical setting may not be immediately applicable. But I think that, hopefully within a few years, that will definitely be a reality. We also believe that it's important now to start to engineer and utilize preclinical models, faithful preclinical models, using both engineered mouse models and patient-derived xenografts to model these alterations. I didn't mention this, but we observed that 80% of these samples also have FLT3 ITD, or FLT3 alterations, most commonly internal tandem duplications. And so, one of the immediate clinical implications is whether FLT3 inhibitors would be specifically effective in this group. And so that's something we would like to test in preclinical models.